Спасибо. So, my dears, finally we get closer to a lecture. I will shorten a little bit this lecture today because we are running out of time, because it is done in not correct place, not correct time. By then I will share you the link on that lecture. Today we're supposed to discuss morphology, structure of bacteria and the bacterial metabolism. But because for already two classes we are studying morphology of bacteria and the structures of bacteria, I won't discuss so much, at least morphology. It's not that difficult to understand. But what I want to I want to demonstrate the place of bacteria in the whole world of living things on our planet. And the thing is that now we may see two ways of uh, classification of all living things on Earth. So these two ways or two approaches are the old approach that we see here, that is five kingdom system of classification, and the next one is three domain system of classification. First, let's take a look at the older one. Here you can see five kingdoms: Planta, fungi or fungi, Animalia, Protista, and Monera. Out of all five kingdoms, you can see the only one with bacteria, that is called Monera. This is the prokaryotic organisms that are unicellular, and the nutrition type is absorptive, so they absorb nutrition from the environment. They may perform photosynthetic or chemosynthetic, and reproduction is asexual. It means without meiosis and mitosis. So, and in Monera or Prokaryota, we may distinguish three basic groups of bacteria: eubacteria or true bacteria, cyanobacteria, that is performed photosynthesis, and archaeobacteria, that are some Asian bacteria that may be different from eubacteria. And the eubacteria are of greatest concern regarding health science and regarding medical microbiology, because mainly pathogenic organisms are found in eubacteria. So, other than prokaryotes, here are protozoa. These are eukaryotic unicellular or multicellular organisms with a nutrition via ingestion, absorption, or photosynthetic. Then animals, that are eukaryotic multicellular organisms that have nutrition via ingestion or absorption. Fungi, that are eukaryotic unicellular multicellular with absorptive type of nutrition similar with that of prokaryotic cells. And plantar, that are eukaryotic multicellular organisms with absorptive or photosynthetic type of nutrition. So, my dears, but this is out of dated system of classification, and the newer one is so called three domain system of classification that divides organism into bacteria, where the only eubacteria or true bacteria are placed into. Then we have a group of so-called archaea, where we have prokaryotic unicellular organisms that lack peptide or glycan cell wall. Generally, these are also bacteria. And the rest of living things, fungi, plants, animalia, and protozoa, are placed into the eukarya domain. where we have eukaryotic, uni or multicellular organisms that are different from bacteria. So, and why this classification is better? Of course, I have shown it in a simplified scheme. But this scheme demonstrates that all those organisms on Earth come from the common ancestral prokaryotic organisms. And then, on particular stage of the development, the different branches had been divided and had onto particular directions. So, and my dears, again, we pay attention at bacteria that have peptidoglycan cell wall and considered to be eukaryotic bacteria. I would prefer if you remember at least this group. And I would like to draw your attention to that and this and to remind you that in none of these classifications viruses are given because these both classifications are given for cellular organisms and viruses are acellular organisms. They are not inserted into those classifications because they are totally different. So they stay apart from all other living things in the world. So, and today we'll be talking about bacteria only. And the most important names of researchers who make some breakthrough in the studies of bacteria are Anthony van Leeuwenhoek, that in 1660s developed the way how to see the very small objects by developing microscopy. Then Louis Pasteur and his work with the uh, diseases of wine. It means he found bacteria that spoil wine. And of course, that's the microbiologist and pathologist, German doctor Robert Koch, that in 1870s did the breakthrough in the way of the diagnosis of infectious diseases. Later on, we'll discuss his very important Koch postulates. So now a few words about the structure of bacteria. You can see that this is the prokaryotic organism. And see here, it doesn't have any well-developed organelle that I've seen, like EPR that are seen, mitochondria that are seen in eukaryotic cells. So it has not developed organelles and some membranous structures that may be seen like mesosomes. So, but then altogether those structures make possible for bacteria to live in the changing environment and to adapt in this environment. So they are quite successful with these not so well developed organelles. So the internal space of bacterium is filled with cytoplasm like in eukaryotic organisms with a colloid system with organic and inorganic solutes in a viscous water solution. So, but then in the cytoplasm you can see distinct areas. The amorphous matrix that contain metabolites, ribosomes, mesosomes, inclusion bodies, so nutrient granules, vacuoles, plasmids, and all others. And the inner so-called nucleoid region, where the DNA is located, that is genome of bacteria. So the cytoplasm is covered and limited with a cytoplasmic membrane that contains both phospholipids and proteins. And you can see here the most important functions of the cytoplasmic membrane, including it participates in very important events like active transportation of the molecules into the cell, energy generation, synthesis of cell wall precursors, because without the cell wall, the cell cannot live, bacteria cannot live, and secretion and enzymes toxins, that is very important for medically important bacteria. Of course, the very important portion of the bacterium is a nucleoid where the bacterial genome is located. And usually the nucleoid of bacteria is made up of the genome consists of single double-stranded circular molecule of the DNA that is approximately have 2,000 to 4,000 genes. And you compare with human that has approximately 100,000 genes. But sometimes it may happen that the some bacteria may have double or diploid genome, so it may have more than one chromosome. For example, it is known for Vibrio cholera. And we believe that before the Vibrio cholera second chromosome had been a plasmid. Then, by obtaining some extra genetic elements, this plasmid had been transformed into the chromosome. So it means it gets as many genes as the chromosome itself. Usually the nucleoid contain no nuclear membrane, no nucleolus, no mitotic spindle, no histones. But my dears, according to the last research work, so they may contain histone-like protein that maintain the structure of the DNA. So apart from the chromosome, and that is a chromosome, we may see also the circular double-stranded DNA structures smaller than chromosome itself that are called plasmids. And those plasmids, and that's a true electron microphotograph of the plasmid, may happen in any type of bacteria without referring to the stain reaction. So in bacteria with thick or thin cell wall. So that is important to understand that plasmids that may replicate independently on the bacterial chromosome are not obligatory or mandatory structures. We may have bacteria without plasmids, but then the importance of the plasmid that they give the selective advantage for those bacteria that keep plasmids. Why? Because, for example, if we compare the stuff without and with plasmid, the Staphylococcus with plasmid may develop resistance against antibiotics, so it may survive in the body, where Staphylococcus without this plasmid will die. So that what means selective advantage. So, in the cytoplasm of bacteria, we may see the very important structure that is called mesosome. It is something, uh, some membrane structure, membranous structure, coil, and invaginated uh, uh, part of the cytoplasmic membrane. See, this is cytoplasmic membrane and this is this invagination. It has multiple functions, by the way. So it usually originates in the place where the transverse septum will be produced during the cell division, and septum will be formed to divide the whole cell in two halves. Then mesosome pulls apart daughter chromosome during the cell division in order to give one chromosome to every daughter cell. And it participates also in electron transportation and energy production instead of mitochondria that is not present in the bacterial cell. I won't talk so much about ribosomes. Everyone knows that these are the universal organelles participate in protein synthesis. So that's why the importance of it cannot be over-evaluated. Then what is typical for bacteria, for some bacteria, is to store some nutrition inside the cell. Usually those uh, organelles with nutrition are called granules, and these are storage areas for nutrition. So, by the way, in some bacteria that can be stained differently from the rest of bacterium, and it will give characteristic appearance to this particular type of bacteria. For example, the volutin granules of Corynebacterium diphtheria is a reservoir of high energy. It is a polymerized metaphosphates which appear as metachromatic inclusion bodies, so metachromatic granules, and they will be stained red with methylene blue dye, and the rest of bacterium will be seen dark violet or blue, and that is called Loeffler staining to reveal these granules. Now we come up to the most important structure of bacteria, that is a cell wall. And you can see, and you know now already, that the cell wall appear in both types. Gram-positive bacteria, here we have thick cell wall, and gram-negative bacteria, sorry, and it consists of the peptidoglycan layer, or briefly it is called murine, and is laying above the inner cell membrane. But then the appearance of this peptidoglycan is a bit different. Then the structure of murine is made up of the polysaccharide or glycan chains, where we have alternated N-acetylmuramic acid and N-acetylglucosamine, like this, right? So they go in one direction. But then the layers of the glycans are connected by the uh, cross-link of the peptides that are bound to the N-acetylmuramic acid glycans. Through this N-acetylmuramic acid, all layers of the glycans are cross-linked with the proteins or peptides. You can see the tetrapeptides and pentaglycine bridges in between tetrapeptides, right? And tetrapeptides are made up of alanine, glutamine, lysine, and alanine. Glucine, okay. So then we may describe it as multi-layered rigid structure. And the structure is very important because it gives shape of bacteria. It protects bacteria from osmotic lysis. And some components of the peptidoglycan may interfere with the human body to cause disease and help bacteria to cause infection in the body. And then, because this part of the bacterial cell is of utmost importance, it also can be affected by some groups of antibiotics. So now you can see that the structure of the cell wall is reflected by stain reaction in gram staining introduced in 1884 by Christian Graham. And those bacteria that have thick cell wall appear violet or dark purple or blue. And those that have thinner cell wall appear pink or red, and we call it gram-negative reaction. And that's something like what you've seen under your microscopes. So that we see due to the single process of staining, we can see the difference between gram-positive bacteria that appear dark violet here and gram-negative that appear red. And again, I would like to demonstrate you a very nice picture, comparatively new picture of the cell wall structure of gram-positive bacteria, where the layers of the peptidoglycan are shown as the violet tubes here, right? And then we can see the lipoteichoic acids anchored in the phospholipid cell membrane and stretched through the peptidoglycan. And also you may see there are teichoic acids that are anchored within the peptidoglycan and also stretches through the peptidoglycan and can be expressed to the surface, right? And that is the cytoplasmic membrane that also can be modified with membrane proteins, porin proteins, and some other protein that participate in the transportation, nutritional bacteria and excretional bacteria. And here you can see that the very important portion of the gram-positive bacterial cell wall are wall teichoic that are anchored in the peptidoglycan and lipoteichoic acid that are anchored in the phospholipid bilayer. And they're important for several reasons. They're important for us because they are immunogenic. So if we get antibodies against these uh, teichoic or lipoteichoic acids. They can be used for identification of unknown bacteria because they are species specific. And they are also, of course, important for bacteria because they influence the membrane permeability, mediate extracellular interaction, provide the cell stability to the in the environment, and also it may act as scaffolds for extracytoplasmic. Then overall this is a structure of gram-negative, gram-positive. Now you see this in the same manner, the structure of the gram-negative cell wall structure. And you can see here that gram-negative is thinner, cell wall is thinner but more complex. So first you see the membrane, inner membrane, that like cytoplasmic membrane. Then periplasmic space where the very few layers of the peptidoglycan are laying, right? Then we have above periplasmic space, we have phospholipid bilayer modified with unique bacterial structure that is called lipopolysaccharide here, right? So, and the lipopolysaccharide here is made up of core polysaccharide or somatic antigens or sugar residues of the core polysaccharide and very important structure that is called lipid A that has very important toxic properties. So lipopolysaccharide accounts for the so-called endotoxic activity of gram-negative bacteria. You can see now here the lipopolysaccharide, right? That is the another name of it is endotoxin. The endotoxin is responsible for many of the features of the disease, and it is responsible for um, uh, inflammation in the body caused by gram-negative bacteria, and as a result of it will be fever, tissue necrosis, drop of blood pressure due to increased permeability of the blood vessels, and endotoxic shock. So if this reaction occurs inside the bloodstream, the so-called Schwarzman reaction may occur. That is disseminated intravascular coagulation that follows the release of large amount of endotoxin into the bloodstream. So here you can see very well the three regions of the lipopolysaccharide. So fatty acids together with O lipid A, then core polysaccharide and the somatic antigen. These are repeated for up to 40 subunits of mannose, arabinose, galactose, rhamnose, and other sugar residues. So the upper part of the lipopolysaccharide is so-called O-somatic antigen. It is made up of up to 25 repeats of 3 to 5 different sugar residues. And it is used to identify certain bacteria because some bacteria, for example, Escherichia gram-negative bacteria, they may have up to 200 different types of somatic antigens that can be identified in clinical samples taken from the patient. And the somatic antigen is exposed on the exterior of the cell. Now, apart from the very important mandatory structures like nucleoid, mesosomes, ribosomes, cytoplasm, cytoplasmic membrane, and cell wall, we may have some additional structures in bacteria that may or may not present in bacteria. And these are non-mandatory additional structures. The capsule, that is a covering outside of the cell wall. Villi, that are hair-like structures that mediate the adhesion to the uh, host body, host cells, but also important in the sharing of genetic information between bacteria. Then endospores that are covered with the several layers that protect DNA, and this is a very resistant form of life. Then plasmid that we discussed are already known mandatory additional structures of bacteria made up of circular double-stranded DNA and flagella used for motions or locomotion function. So the important thing is the capsule that lies outside the cell wall, and it is composed of polysaccharides usually, and in Bacillus anthracis it is made up of D-glutamic acid, so it has polypeptide nature. So the capsule is a determinant of the virulence of many bacteria, and it limits the ability of macrophages to engulf and kill bacteria. So they also may play as a sticky layer. They may play an important role in the adhesion of bacteria to human tissues. Then usually the capsules are immunogenic, so they may evoke the immune response against capsule antigen. So then the antibodies against the particular capsule antigens can be used in identification of bacteria. Moreover, some capsules may act as the antigens used in vaccination. For example, vaccination against staphylococcus, against streptococci, and some other cocci. This specialized structure is also flagellum that participate in moving bacteria. And the flagellum act as a propeller. It is composed of many subunits of so-called flagellin, a single protein arranged in several interwound chains. And then the energy for movement is usually taken from the proton motive force, derived from the passage of the ions across the cytoplasmic membrane and provided by adenosine triphosphate ATP molecules. So, flagella are important. Sometimes they even recognized as a virulence factor. For example, the flagellum vibrio cholera is recognized as a factor that allow or mediate the movement of bacteria towards the receptors on the mucous membranes. So the flagella can be located in several ways. You can see monotrichous, trichous, lophotrichous, amphi on both sides, and peritrichous. And here you can see the examples of bacteria with one uh, flagellum, that is Vibrio cholera. Uh, Amphitrich bacteria, that is a Campylobacter. And here we have the peritrichid bacteria that's regularly, for example, Escherichia coli. So, and Lophotrichous bacteria, usually this is Campylobacter. So now the very important structure of the bacterium is the spore. Here you can see the result of Gram staining, and you can see that spore itself, see here, is not really stained by Gram staining. It looks like a hole in the rod, right? Here you can see gram-positive rods with spores. And these are spores. So we have just two genera that may produce spores. These are Clostridium and Bacillus. And the spores are produced in non-favorable conditions. See, for example, so if we see the free endospore, when we place it in good conditions, it will germinate, and the process is called germination into vegetative cell. Then after some while, vegetative cells divide, grow, but then if conditions will be worse, so they start cell division and divide into portions in one of which the developing of the spore coat will be seen. And then this will be developed around the genetic material with very few ribosomes. This process is called sporulation. As a result of sporulation, the cell is produced, protected from outside. Then the rest of the rod undergoes autolysis. So it means the remains of the vegetative cells here will be dissolved or disappeared. So, and then finally we see the only endospore. That is called sporulation. So, sporogenic bacteria have a life cycle with alternating sporulation and germination. And here you can see the endospore structure, and it is called endospore because the spore is produced by concentration of the cell cover of the spore coverings inside the cell. So that's why we call it endospore. This is a process of forming spores, and this is the endospore itself. So, and then the endospore usually covered with exosporium, then with a spore coat resistant to the chemicals, then with cortex, see, the cortex that rests beneath the spore coat. Then we have the core cell wall, and that is the remain of the bacterial cell wall. And it usually surrounds the core CR with a nucleoid. And here you can see the moment of spore germination. So now a few words about the nutrition of bacteria and how we manage to culture bacteria in the laboratory. So, of course, for growth and multiplication of bacteria, we need a lot of things. But then, mainly, we need what we need for any living thing growing in the world, right? So we need water. As for other organic organisms, we use carbon, and carbon is one of the most important elements. So those are called macro elements, right? And these are also oxygen, hydrogen, nitrogen, sulfur, inorganic salts, and altogether we can see 10 out of them here. So, but then bacteria also require micronutrients, so-called, that can be part of the enzymes or cofactors. And in addition, they need energy. So regarding the nutritional types, bacteria can be classified into autotrophs and heterotrophs. Mainly pathogenic bacteria are heterotrophs, but not autotrophs. So what is the difference? Autotrophs use carbon dioxide as a sole source of principal source over the biosynthetic of the structures. Then heterotrophs usually use reduced preformed organic molecules produced by some other organisms. So then they also can be classified according to the energy source they use. For example, phototrophs, they use sunlight, the energy over the visible light of spectrum, right? Then the second type is chemotrophs that perform energy due to oxidation of organic or inorganic compounds. And according to the electron source, the chemotrophs can be subdivided into lithotrophs that reduce inorganic molecules and organotrophs that usually reduce organic molecules. So, according to the temperature requirements, bacteria may be classified into the thermophils, psychrophilic bacteria, and mesophilic. We as medical microbiologists are interested in mesophiles because mainly all pathogenic bacteria will grow in between 20 to 40 degrees Celsius, and the optimal temperature is 37 degrees Celsius. The psychrophilic, they grow below 20, and thermophils grow at the temperature higher than 55 to 80. Then the pressure is not so important unless it is high. So certain bacteria, usually bacteria cannot stand high osmotic pressure. And the only halophilic bacteria or staphylococcus with very thick peptidoglycan layer may stand the presence of the up to 10% of the sodium chloride. And also the optimal temperature for bacteria is usually neutral or slightly alkaline. But of course, we always have some exceptions. For example, Vibrio cholera would like to grow at the temperature, oh, sorry, at the pH very basic, up to 9.6 pH. That is unusual. But then from time to time, some bacteria may demonstrate this. So then bacteria can be classified also according to the oxygen requirements. So we may have some wider classification, but then usually we classified it into four groups. And those four groups are obligate aerobic bacteria that live in the presence of the oxygen only because they use oxygen and the ultimate acceptor of the oxygen, of the electrons. So that another one is obligate anaerobic bacteria like Clostridium tetani. So, because they cannot grow in the presence of the oxygen, because as soon as it starts, the free radicals will be accumulated in the bacterial body, and so the bacteria will be destroyed. So, by these free radicals. And that also happened in the obligate aerobes, but obligate aerobes may produce enzymes which split toxic products, for example, hydrogen peroxide, by superoxide dismutase, and then they inactivate the free radicals. Facultative anaerobes and microaerophils are somewhere in between obligate aerobes and obligate anaerobes. So how these bacteria derive energy? First, we start with aerobic bacteria. Aerobes generate the energy and intermediates only through oxidation, using atmospheric oxygen as the ultimate hydrogen acceptor and cytochrome respiration. The process can be, so as usual in biochemistry, we divide the process into several blocks. So the first block of reactions, yeah, and I'm not going to demonstrate you those reactions. So the first block is glycolysis. The glucose is taken up by the cell and broken down via glycolysis. So that means glyco sugar and lysis split. So very simply, glycolysis uses enzymes to split glucose molecules into the pyruvate. During the breakdown of glucose, electrons are liberated and used to form NADH or nicotinamide adenine dinucleotide, as an important electron carrier in the cell. Then the second block is the citric acid cycle. That is a series of enzymes used to break down pyruvate all the way to carbon dioxide. This cycle oxidizes the pyruvate, capturing its electrons and depositing the electrons on NAD to form NADH. The NADH formed during glycolysis and the citric acid cycle connects to the next block in our pathway, that is electron transport chain. And this is the final stage of respiration. So when ATP is formed through a series of electron transfer reactions within the cytoplasmic membrane that drive the oxidative phosphorylation of ADP to ATP. And bacteria use various flavins, cytochromes, and non-Hem iron components as well as multiple cytochrome oxidases for this process. So the electron transport system is located on the cell membrane, as we said, and is composed of the cytochrome enzymes, lipid cofactors such as vitamin K, coenzyme Q, and as a result, 38 molecules of FTP will be generated. Then autotrophy is a unique form of metabolism found only in bacteria. When inorganic compounds are oxidized directly without using sunlight to yield energy. And this is ammonium, the nitrate oxide, and ferrum, and then the others. This metabolic mode also requires energy for carbon dioxide reduction, like photosynthesis, but no lipid-mediated processes are involved. This metabolic mode has also been called chemotrophy, chemoautotrophy, or chemolithotrophy. So some bacteria exhibit a unique mode of respiration called anaerobic respiration. These heterotrophic bacteria that will not grow anaerobically unless a specific chemical component, which serves as a terminal electron acceptor, is added to the medium. Among these electron acceptors are nitric oxide, sulfur oxide, the organic compounds fumarate, and carbon dioxide. Bacteria requiring one of these compounds or components for anaerobic growth are said to be anaerobic respirers. Anaerobic metabolism generates from 2 to 12 ATP molecules depending on the particular cycle, because there are a lot of the electron acceptors, so it depends on the cycle. Bacteria may use many cycles. But then after the oxidation of one glucose molecule, from 2 to 12 ATPs are produced depending on the substrate. So the organic substrate act as the electron acceptors to recycle the reduced form of NADH to NAD, so like this. And this process is known as fermentation, and many bacteria are identified on the basis of their fermentive end products because they are highly characteristic to bacteria that produces it. Of course, this way of anaerobic substrate level phosphorylation is much faster than that of the aerobic respiration. But of course, it yields less energy from one molecule of glucose. But then sometimes it may give us a lot of useful products, such as lactic acid, alcohols, gases, but then they are characteristic of the bacterial species. So how about the facultative anaerobic bacteria? Are they aerobes or anaerobes? My dears, they are aerobic bacteria. And they grow much better and faster in the presence of the oxygen because they love oxygen. And because this pathway is 19 times usually more effective to compare with anaerobic respiration. But then if you place them in anaerobic condition, they will also develop energy and grow very well. Now let's talk about the growth and culture of bacteria. And you can see here that the major instrument that we use is bacteriological loop. With this bacteriological loop, you can see a wire that form loops at the very top of the of the bacteriological loop. Then we have a handle and the whole metallic core, right? So where the wire is fixed with. Okay, what do we need to mention regarding culture of bacteria? A pure bacterial culture, what does it mean, pure bacterial culture? It means this culture consists of one single type of bacteria. And usually, bacterial culture remains essential for the study of the bacterial virulence, antibiotic sensitivity, genome sequencing, in order to facilitate the understanding and treatment of caused disease. As had been proposed by Robert Koch. A pure culture is the foundation of all research in infection diseases. The first isolation of the bacterium in pure culture enabled the design of experimental model to analyze virulence and to compete or to complete Koch criteria, thereby establishing a link between microorganism and infectious disease. And this is very important in diagnosis. The postulates were formulated by Robert Koch and published in 1890. Koch applied the postulates to establish the etiology of anthrax and tuberculosis, but then later on they have been generalized, those postulates, to all other diseases. What are the Koch postulates? As following. The microorganisms must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. This is the first one in a system of proving the causative agent of the disease. Second statement. The microorganism must be isolated from a diseased organism and grown in the pure culture. It means it should be isolated from all other bacteria found in that body and grown separately from them in pure culture. It means solo. Then the third statement. The cultured microorganism should cause disease when introduced into a healthy organism. Of course, as a healthy organism, the laboratory animals had been used and are in use now to demonstrate the ability of that organism to grow there and to cause disease. And finally, number 4. The microorganism must be re-isolated from the inoculated diseased experimental host and identified as being identical to the original specific causative agent found in the human body. That is a system of proving the microbes as causative agents of the disease. Of course, later on we improve it a little bit. For example, we add number 5. So what is it? Because we found new methods of proving the causative agent of the disease. And for example, the serological test that may help us to find antigen of bacteria or that make us to find the antibodies against particular microbe. Then finding of this either antigen or antibodies will be also enough to prove the causative agent of the disease. Then now the question is how bacteria grow. We have to understand that they may grow in size and in numbers. When bacteria grow in size, that accumulate the enzymes, nutrition inside. And when bacteria make twice the size of initial bacteria, only then they may start dividing. And bacteria divide uh, by binary fission, and that is the way of bacterial propagation. A cell just need to grow to, but to remain viable and competitive, a bacteria must divide at the right time, in the right place, and must provide each offspring with a complete copy of its essential genetic material. So you can see the process right here of dividing, producing the septa in between two daughter cells. Before binary fission occurs, the cell must copy its DNA and segregate these copies to the opposite ends of the cell. Then the many types, around of 10 different types of proteins that comprise the cell division machinery, assemble at the future division site, and that is a future division site. A key component of this machinery is the protein FTSZ. Protein monomers of FTZ assemble into a ring-like structure in the center of the cell. You can see it here. And then the septum is formed through the inward growth of cytoplasmic membrane and cell wall material that invaginates from the opposite directions and the central plane of the cell. Other components of the division apparatus then assemble as FTSZ ring. This machinery is positioned so that division splits the cytoplasm and does not damage DNA in the process of the division. As division occurs, the cytoplasm is cleaved into two, and in many bacteria, the new cell wall is synthesized at the place when mesosome occurs, because that is important. So now, then we know that how bacteria divide, so we may take at the bacterial population growth. So usually if you would like to culture them in the laboratory, they can grow in three main forms. The first one is seen here. So bacteria may grow by development of the colonies or so-called clones of bacteria. And what is a colony? It is a macroscopic product. What does it mean, macroscopic? Visible by naked eye. And usually it occurs as a result of 20 to 30 cell divisions of a single cell. My dears, when we try to perform growth, we would like to see the colony well separated from the others. Why? Because the only then we may expect that this colony, well separated from the others, is made up out of one single bacterium. If we take the colonies like this or like that, we can see that they are fused together. Most probably those colonies can be used as a source of the clones of bacteria and cannot be recognized as a clone because many different types of bacteria may be mixed up here. But that well-separated colony is recognized as a clone if we see under the microscope the smear prepared from this colony that it is made up of one single type of bacteria. The only then it is considered as a clone and can be used further for pure culture isolation. So then the colonies are formed on the surface of the or inside in the depth of the solid medium. The colonies are never formed in the liquid medium. Okay? Then the second way of the growth of bacteria is the growth in the broth culture. So the colonies are not formed. And in the broth culture, bacteria may grow also in the different manner. So most commonly, that is non-inoculated medium. Most commonly, bacteria form a universal uniform turbidity in the medium. Here, the medium here is clear, here is not clear. It is turbid. That is a sign of bacterial growth there. And usually, if we have this turbid suspension, we may have 100 to 1,000 million cells per milliliter. That's a lot. Then sometimes some bacteria, for example, Vibrio cholera, that are really aerobics. They are obligatory aerobic bacteria. They may form a sort of surface growth. We see the pellicle formed on the surface of the broth medium. So that is pellicle growth. And some bacteria may, during the growth, they form the very long fibers like streptobacilli or streptococci. Then usually these long filaments made up of many bacteria. They fall down the bottom of the uh, tube with the culture broth. So we call it sedimental growth because the sediment is formed. That's how in what uh, uh, how the patterns of bacterial growth in the liquid broth look like. And finally, bacteria may grow by forming biofilms on the substratum. So, and that's most probably how bacteria grow in the human body. Because usually this biofilm is made up not of one type of bacteria, but of several types of bacteria. Usually biofilm is of 300 to 400 micrometers thick, and they can be produced on the inert surface or artificial surface or natural things or whatever. And usually this way, this growth can be divided into stages. So first substrate will be preconditioning by ambient molecules like proteins. That's quite often. Then subcells may be deposited on the pre-prepared area, and the cells may finally absorb. Some will be desorbed from the substrate, but some they stay there. And they produce quorum sensing like cell-to-cell signaling, and bacteria start producing so-called exopolymer, and that is something like the cell wall material, but that is secreted to outside. This exopolymer fills the gaps in between bacteria and makes substrate more uh, suitable for the bacterial growth. Then we can see connective and diffuse transport of the oxygen and nutrition. And that's very important because different bacteria here may differently perform metabolism, and those bacteria that are anaerobic will be seen deeper in the exopolymeric structure, and they will not have the access to the oxygen. And those that are aerobic will be on the surface usually. And then they perform replication and growth, and secretion of the polysaccharide matrix again will thicken the layer and fill the gaps in between bacteria. And then how it exists. Some of them may detach from this biofilm, and this soften will allow this bacteria to be transported in the body and then to cause biofilm formation in distant areas. That's how it grows. Then, what types of cultures usually we use and how bacteria may grow in the uh, laboratory, in the cultures. So when we may have two systems for their growth, closed system and open system. What do we call a closed system or so-called batch culture? When then bacteria are when medium is inoculated and then we add nothing to this bacteria. So this is a closed system. We add nothing and get out of there nothing. So and if we consider these conditions, usually bacterial population uh, may follow the several phases in its growth. Lag phase, when bacteria grow in size and get used to the new conditions. Then, logarithmic or exponential growth phase, when the cells enlarged in size start quickly dividing. Then it reaches the stationary phase where the number of bacteria won't change so much. Bacteria start continue dividing, but also some start dying. So that's why the number of bacteria will stay stationary without any changes. And then eventually, when we have the decline of the uh, nutrition and the accumulation of some toxic compounds, the metabolites, the result of the metabolic activity of bacteria. So then they start dying. So we have a logarithmic phase of decline. And it may reach the complete death of bacteria. Then that what we have, that what we have in a petri dish usually. Then the next one is the continuous culture. When bacteria grown, the fresh medium flows into the fermenter continuously, and part of the medium in the reactor is withdrawn from the fermenter at the same flow rate of the inlet flow. Then the rate of addition of the fresh medium determines the rate of growth. The bacterial culture can be grown and maintained at relatively constant conditions in the stationary phase, and in fact, this plateau stationary phase may last for a very long while. Not really forever, as you understand, but then for a very long while. So then this is the scheme how it can be performed. So that is the called bioreactor chemostat. Once inoculated with bacteria with shaken, usually in the broth culture, these bacteria start growing. So through the vacuum pump, the nutrient supply occur with the speed with which bacteria will be, or the used up medium together with bacteria will be taken out and collected for particular product that we need to produce. Then through the pumps, we may take the samples to perform pH control and whatever nutrition control. And then we may add the sterile source of oxygen and remove the gases if we need them anymore. So that's how it occurs in mini reactor. So when the volume is very small. You can see the control tower with the electrode to measure pH continuously, and this is the pump to pump in the through inlet the uh, new nutrients, and through inlet the used medium together with some product of interest will be taken out through outlet. So, few words about the isolation of bacteria. What approaches do we use in isolation of bacteria? Usually, we are talking about types of the culture that are used in isolation. That, of course, the agar plate. So, these agar poured in petri dish. And usually, the agar plate is used to separate bacteria from the sample taken from the patient. To disperse these bacteria, so to see somewhere the well-separated colony. Then we may have the agar deep tube that is used to culture to the growth of anaerobic bacteria. Then we have a liquid medium, the culture broth, to increase the number of bacteria dramatically because Bacteria grow better in the liquid medium than in the solid medium. And also we may see agar slant, and this culture is used to keep the pure culture of bacteria. But initially, of course, a clinical sample of material is taken from a human, such as throat swab or urine specimen or blood can be taken, whatever. And usually the specimen taken from the patient is placed on the petri dish to separate all bacteria in the specimen, to see them separately. And then after this, we usually put the plate into the incubator for an overnight incubation. And in the morning, we are going to see well-separated colony on agar slant to get somewhere in the plate a pure colony, as a source of pure growth of bacteria. But before we use this colony as a pure growth of bacteria, we have to demonstrate microscopically that in a smear taken from this colony and stained, for example, by Gram staining, we see one single type of bacteria. So if we see one single type of bacteria, so it means we managed to get them well separated. So then we pick a colony from the streak culture, check whether it is pure or not, and then we perform the inoculation of the straw culture or so-called slug. And it is performed to get a pure growth of bacteria. And it is used because we use tubes with a narrow neck, and it is easy to keep it not contaminated with other bacteria. Much easier than in a petri dish. And then we supposed to follow the identification algorithm of bacteria, and usually you may take a look at it. It is performed with the help of biochemical tests because the morphological features are not enough. So then what do we use for identification of bacteria? So when we get the culture already, the second thing is to identify these bacteria. So we identified it by looking at microscopic morphology, that what we see under the microscope. Then we can see the colonies, how they appear on the solid medium. Then we perform some biochemical tests to identify what kind of enzymes can be produced by this bacterium. And then we may perform a sort of studying of pathogenic properties, biotyping, serotyping, phage typing, and the others. That what we see as a result of microscopic morphology of bacteria. See how much information we may get from here. So the shape of bacteria, then the presence of the spores, the arrangement of bacteria, then they can be seen here, right? Then if we are going to culture fungi, that's not so big difference in the identification of the fungi and bacteria. We also may see the shape of the fungi, and microscopy is more important for fungi identification than for bacterial identification, because fungi also may form some reproductive structures that are specific for particular types of bacteria, and even fungal spores may be used for identification. Then we can see the cultural characteristic of bacteria, that is bacterial colony morphology. That's how the bacterial colonies may appear, and that we see by eye. And then we may see some of the characteristic of these colonies. They can be flat or raised. They can be entire margins of the colony or filamentous, and the form of colony may be pantiform, circular, filamentous, irregular, whatever. So, and then these are the morphology, this is the macroscopic morphology of the fungal colonies. And then we perform biochemical tests. Then we may perform additional tests, serotyping or genotyping that is used for identification of bacteria. Then we perform antibiotic sensitivity testing. But these are additional tests. By the moment when we perform antibiotic sensitivity testing, we know already the name of bacteria according binomial nomenclature. What does it mean, identification of bacteria? That's identifying the name of bacteria, for example, as how do we call it? Staphylococcus aureus or Streptococcus pyogenes. That is the name of the genus and the name of the species. And then we also may divide the species of bacteria into the strains. What is a strain? Strain is made of the up of the organism that are descendants of one single isolation in pure culture. Usually it originates from one single well-separated pure colony. This is the population of the organism with characteristics that distinguish these serogroups of a species from other serogroups of the subgroups of the same species. So within one species, we may have many strains of bacteria because the strain is the culture isolated from the particular source of bacteria at the particular time and identified as particular species. So then, bacteria belong to Monera, and then we also may identify their class, order, family, genus, species, subspecies, and strain. But of course, the basic taxonomic group is species. A bacterial species may be regarded as a collection of strains that share many features in common and differ considerably from other strains. Species is the basic taxonomic group. It can be designated as a collection of strains that have a similar guanidine-cytosine contact and at least 70% DNA similarity as determined by nucleic acid hybridization experiments. My dears, with this I would like to finish, and the link will be given in the microbiology department VK site. And I hope usually next time we won't have any more troubles with this. So as soon as you would like, you expect lecture, please take a look at the department site in Wiki to see the link of these lectures. With this, goodbye, my dears. See you next time.